Okay, ladies and gentlemen, welcome to the Royal Society this evening to the Leeuwenhoek lecture. My name is Jean Thomas and I'm the Biological Secretary. And I'm asked to remind you that you should please switch off your mobile phones before we start because the lecture is being recorded and webcast. And it's a great pleasure to welcome you all here in this 350th year of the Royal Society since its establishment. It's a very special year. If you've been watching the slides, you'll see that events have been going on for 12 months and are still going on right up until the 30th of November in London and all over the country. And the culmination of this will be the Summer Science Exhibition, the end of June, beginning of July in the South Bank, which is open to everyone. There are also many events in collaboration with museums and galleries, and local heroes programs all over the country where local heroes are celebrated in their hometowns or in local museums and institutions. And there's also a book edited by Bill Bryson with 20 interesting chapters called, I think, Seeing Further. And that's on sale, probably rapidly running out, so grab your copy now if you want. And this is to mark the 350th anniversary year. So that's the business out of the way. I'm charged with reminding people of this every time we have a lecture. So to the business of the evening, the Leeuwenhoek lecture is named, as you all probably know, after the great Dutch microscopist Anton van Leeuwenhoek, and it was established to recognize excellence in the field of microbiology. It now recognizes excellence in bacteriology, virology, mycology, parasitology, and in microscopy, Leeuwenhoek's original sort of subject. It was originally held annually, and it's now given triennially. It was endowed by the George Gabb Fund, which was founded in 1948 by a bequest of £1,000 from Mr. George Gabb. So that's the lecture, and the lecturer, our distinguished visitor this evening, is Professor Robert Webster, who is professor in the Division of Virology, Department of Infectious Diseases at St. Jude's Children's Hospital in Memphis, Tennessee, and he holds the Rosemary Thomas Chair there. He hails from New Zealand, and he received his BSc and MSc in microbiology from Otago University. He then migrated to Australia, to the Australian National University, the ANU, where he gained his PhD in 1962. He then moved again, this time to the United States, to the Department of Epidemiology at the University of Michigan, Ann Arbor, where he spent the next 2 years as a Fulbright scholar working on influenza. And Dr. Webster is internationally known for his subsequent work over the decades on influenza and is an extremely deserving recipient of the Leeuwenhoek Award this year. Professor Webster's interests include the emergence and control of influenza viruses, viral immunology, the structure and function of influenza virus proteins, and the development of new vaccines and antivirals. The main focus of his research is the importance of influenza viruses in wild aquatic birds as a major reservoir of flu viruses, and their role in the evolution of new pandemic strains for humans and lower animals, which you will all recognize, I'm sure, as being highly topical. He's the author of over 500 original articles and reviews on flu viruses, and he's trained many scientists who now contribute to our understanding of the evolution and pathogenesis of influenza. He also established the Center of Excellence program for flu research and surveillance at St. Jude's Children's Research Hospital. Professor Webster is a Fellow of the Royal Society, elected in 1989, and of the National Academy of Sciences of the United States, elected in 1998. And it's my very great pleasure now to present Professor Robert Webster to deliver this evening's lecture with this wonderful title, Pandemic Influenza, One Flew Over the Cuckoo's Nest. Thank you very much.
Good evening. Professor Thomas, staff of the Royal Society, fellows of the Royal Society, ladies and gentlemen. I'm deeply honored to be invited to present the 2010 Leeuwenhoek lecture on the 350th anniversary of the Royal Society. And since this is the period of the World Cup, I thought we should begin with some thoughts about the World Cup. And my first slide shows my evaluation of how science is doing and how influenza is doing. At this stage, we see that the influenza virus is 4 goals up and seems to be gaining, while the scientific crew haven't scored recently. The strategy of the influenza virus, of course, is to reassort, you develop drug resistance, and spread like crazy. Let me introduce the captain of the team, the influenza virus. The strategy is to change the jersey as often as possible to the spike proteins, the hemagglutinin, the neuraminidase. We know all about the structure of these, but the virus just keeps changing the patterns so the referee doesn't really know what's going on. Additionally, the captain has only 8 members, not 11 in his team, but he can pull in a whole new cadre of players from all of the wild birds of the world, overnight, as it were. So these are the strategies that make this captain enormously variable. And that's the strategy of influenza, of mutations and reassortments. If we put 2 of these different influenza viruses into a cell, in one round of mating, they produce 256 different offsprings. So that's the strategy the captain uses. And so why have the scientists failed? They've been planning pandemic preparedness for the past decade or more. The experts predicted H5N1, so-called bird flu. But nature sent H1N1, related to 1918 Spanish influenza. And this terrified the scientists of the world. The 1918 Spanish influenza occurred during and at the end of World War I. Why Spanish influenza? Because neither the Allies or the German side wanted to acknowledge that influenza was going on in the troops. And so the first outbreaks was described in Spain, and so it became Spanish influenza. It turned out to be America's secret weapon. The Americans didn't realize what they were doing, but they used biological warfare to finish World War I. The German generals wrote in their memoirs that it wasn't the tanks, it wasn't the guns, it was the biological warfare that spread from the Allied side across the trenches were so close. And this virus killed somewhere between 50 and 100 million people in the world. And so when H1N1 appeared in 2009, we can understand why the scientific community was so concerned. The 1918 virus came from somewhere in the heartland of the United States. And it showed up in Camp Funston in Kansas. That's when the soldiers started to die that was kept under wraps in the United States. Then those soldiers were put onto ships to be sent to Europe, and crushed onto ships. They died like flies. And the actual numbers that died is really not known because it didn't get into the records. This is one of the earlier ships that came, the plague ship, the terrible ship, the USS Leviathan. In the 29th of September 1918, 9,000 doughboys, American soldiers, left for France. 2,000 were ill and 91 died. That's what was told about. The suggestion was made to the President of the United States, Wilson at the time, to stop sending the troops. There were so many dying. When Leviathan was referred to as a ship from hell, there was blood and gore everywhere. When the ship reached Brest on the 9th of November, it took hours and hours and 4 or more miles of carriers to take the sick and the dead off the ship. So this didn't bring the virus to Europe. Probably one of the earlier ships did. And so now I want to switch to New Zealand. If you note the time that happened, in New Zealand we know exactly when this virus came in. The Prime Minister, Massey, had been to Canada on a meeting to do with the end of the war. And he came back to Auckland. And on board the ship, there were 3 or 4 cases of flu. They didn't quarantine the Niagara since the Prime Minister was on board. And between the 12th of October and the 1st of November, Auckland became a city of the dead. And by November the 12th, there were 2 death trains a day leaving Auckland. It's a remarkable similarity in time between what happened in New Zealand, what happened in England, and what happened in Alaska. In England, the troops brought the viruses back and went through the main railway stations and spread out from there. Similar situation, the dead couldn't be buried. Coffins built up in the streets. The next case I want to deal with was the outbreak in Brevig Mission, Alaska. The sailing ship had come into Nome, and there was a total quarantine. There was no influenza on board the ship as far as they could tell. But when they put the letters on the dog sleds, the dog sled driver took sick on the way to Brevig, and unfortunately the dogs pulled him into Brevig Mission. And between November the 15th and the 20th, 72 of 80 people died and were buried in the permafrost. Interesting that children survived. This virus, we always worried about what made it so pathogenic. Why was it so pathogenic? And since we didn't have freezers at the time, the decision was made to go and dig up remains, as several of us have tried to do. Johan Hultin went back to Brevig Mission and dug up some of the remains. Jeffrey Taubenberger, over many years, patiently sequenced and obtained the total sequence of the virus. But having the total sequence still told us nothing about the pathogenesis of the virus. So it was a case of remaking it. Should we bring back a virus like this from the grave and remake it? And the decision was made, yes, it should be done, but very carefully under controlled conditions. Several scientists have remade it. My colleague Kawoka and his staff, Kabosa, did the studies in the high containment facility in Canada and reconstructed the 1918 and compared it with a then circulating H1N1 from Karasaki. And the macaques, within a few days, showed very high virus loads, aberrant cytokine activation, severe primary pneumonia, and death. In fact, the macaques had to be put down. And so this resurrected virus was enormously pathogenic. And so that's what worried us when H1N1 reappeared in 2009. Where does it come from? Where did this virus, where did they come from? And I think we all know it's the aquatic birds of the world, and they move through intermediate hosts into humans. The residents of all of the influenza viruses are the aquatic wild birds of the world. There are 16 families of hemagglutinin, 9 neuraminidase subtypes, and they all exist in the wild birds where they cause no disease. No disease, and the virus replicates mainly in the intestinal tract and is shed fecally, orally. Wonderful way of transmitting viruses in the world. I want to turn to where most of the information came from. My colleague Graham Laver and I were walking on a beach on the coast of Australia with many dead birds. Graham went to the head of department and said, we should study influenza on the barrier reef. And Gordon Ada said, you've got to be crazy. We're not paying you to go to the barrier reef to have holidays. But Graham went to the World Health Organization, and the World Health Organization gave him $500. So he went to the barrier reef to sample birds. The first year, he sampled the short-tailed shearwater, Puffinus pacificus, and found antibodies in the short-tailed shearwater, similar, that identified the human influenza viruses of that time. And the World Health Organization gave a little money, so he went back following years, and from the noddy terns, isolated several viruses, one in particular, A. tern Australia, G50C 75, H11N9. And I'll come to the importance of that virus just in a moment. These birds migrate around the world, mainly in a north-south direction, but the short-tailed shearwater migrates right around the Pacific, comes back and lays its eggs and breeds off the Australian-New Zealand coastline. And so Graham studies, and that virus isolated from the noddy tern, he isolated the neuraminidase from it, crystallized it, and based on the structure of the neuraminidase, we now have Tamiflu, in conjunction with many other people, of course. But essentially, he went from the barrier reef to the bedside. So how do we move an influenza virus from the wild birds into humans? With all of those influenza viruses out there, we might imagine it would occur fairly frequently. But in fact, it's a very, very rare occurrence. And if we look at some of the properties, we will understand. The influenza virus in birds replicates 42 degrees, internally in the intestine. 42 degrees, mammals 37. Site of replication, intestine, respiratory. Mode of spread, fecal waterborne, respiratory. And the receptor specificity, the binding sites, recognize terminal sialic acid of the 2,3 variety and the mammalian 2,6 variety. So all of these changes have to be built up for an influenza virus to make it from the wild birds across into humans. And the pig seems to be what we recognize as at least one of the mixing vessels, because the temperature of the pig is about 39. The site of replication is mainly respiratory, but it has receptors for both kinds and is mainly respiratory. So the pig tends to be the intermediate host. And the pig and the pandemics of the past century, the role of birds, the 1918 Spanish, we're now not quite sure. We used to think that all of the segments of the virus came from birds, but we now, my colleagues in Hong Kong, believe it was also one of these reassortants between humans, avians, and maybe pigs. We know that the pig was certainly involved in the genesis of the Hong Kong H3N2. We believe that it was involved in the genesis of the Asian pandemics. So we had 3 pandemics in the past century, and H1N1 reappeared. Then along comes H1N1 again out of Mexico in April. I want to walk you through where and how this virus occurred. We now know that the influenza viruses in wild birds quite frequently go into pigs, but most times it's benign, doesn't go anywhere, doesn't spread from pig to pig. But the H1N1 viruses have a tendency to go into pigs and stay there. 1918 Spanish influenza, and then in Italy in 79 in Europe, an H1N1 virus. Again, in China, it didn't spread far. Other viruses that go into pigs, and so it's a frequent occurrence. Then in humans, many of the human viruses will go successfully in the pig. The events in the genesis of the 2009 pandemic, if we look at the 1918 Spanish influenza, it got pieces presumably from the wild bird, from the human, and the pig. And that virus remained in pigs from 1918 to 1998, and was very, very mild. In fact, it became more and more benign, almost disappeared from pigs in significance for disease. Then in 1998, after nearly a century in the pig, something happened. This virus started to reassort, and one of the first reassortants we detected was a reassortance between the virus in pigs, the classical 8 segments from the pig virus with a human virus, to give a double reassortant. This wasn't very successful, but we did isolate it from the pig. Then in Europe, another family of H1N1 viruses developed, and described by Pensrant in 79. Another H1N1 from wild birds went into pigs. And again, after some time, it mated with the human virus to give what we call a double reassortant. Then back to United States, again in 98. Something happened in 1998, and we still don't fully understand. Nearly 100 years it stayed stable, didn't do anything. After 1998, this virus mates with anything it can find. One of the first 3 that we have, we call the triple reassortant. It got 3 of its segments from the human, 3 from pigs, and 2 back from the wild birds. And so this is one of the important precursors of the pandemic. It took something like 100 years to get its pieces, its team in order, if you wish. Then in April 2009, the pig reassortant from Europe and the triple reassortant from United States that had already mated with something else, with one of the swine viruses, all got together to give the pandemic H1N1 that we now know caused the most recent pandemic. Somewhere in the Americas, presumably in Central or South America, where no surveillance was going on. And so panic set in. April 2009, swine flu and the connection back to 1918. The desperate need for a vaccine rapidly, which we really didn't achieve. This virus, between April and November, essentially spread across the whole world. It's not only spread, and by November it had killed something like 6,000 people. Current death toll is at just over 18,000. The virus likes to spread. It spread to pigs, back to pigs in many countries, to turkeys, to cats, to dogs, to ferrets. And so this is a real spreader, and it's got many hosts to get mixed up in. So what are we doing about surveillance in pigs? The best surveillance in pigs in the world is going on with my colleagues in Hong Kong. Since 1998, after H5N1 was detected in Asia, surveillance has gone on in pigs continuously, month by month. And the current situation is this pandemic virus is being picked up in pigs. Standard H1 is being picked up in pigs. The H3N2 virus has disappeared, and they have picked up in January a novel virus that's going to be described in Nature next week, actually. So I can't really describe it other than to tell you that, again, that it's made a reassortant. And so this virus that's currently extremely mild in humans is still mating and picking up new genes. This studies on the molecular clock of these viruses suggest that they've been out and about for between 9 and 17 years. So scientifically, we missed the boat. The precursors of that virus was in the pig population for something like 17 years, and we didn't know it. We were not doing surveillance in the right place in healthy pigs of the world. And so it circulated undetected for over a decade, and described by the folks in Hong Kong. But to turn to the other influenza, the one that we spent a great deal of time planning for, the H5N1. The H5N1 virus emerged from the wild bird population somewhere in 1996 in southern China. It spread to over 60 countries in Eurasia. Over 500 million poultry have been destroyed. And currently, nearly 500 human cases have occurred, with 294 deaths. The saving grace for this virus is it doesn't spread human to human. It kills something like 2/3 of the people infected, but fortunately it doesn't spread. But it hasn't gone away. It's still alive and well in part of the world. I want to walk through the genesis of that virus. In 1996, a single child died in May 1996. And no one gave it a lot of attention. In fact, it wasn't really characterized until August in Holland. And then November, people started to die, and the emergency was declared in Hong Kong that H5N1 really was a problem. This virus came through the live poultry market, brought in by ducks and the chickens And as my colleagues in Hong Kong point out, the chickens end their lives there, but the virus doesn't. It goes back and spreads. And so what they'd done in Hong Kong, in fact, was they'd created high-rise live markets. This is this pink building here, so they don't sell a few chickens off the back of a truck or off a wheelbarrow. They have sophisticated market system. When you walk in the door of that market, you meet the chickens in their all ready for the pot, as it were. And in addition, all of the kinds of birds. The quail behaves a bit like the pig in being an intermediate host, and pigeons and all of the different, there's pheasants here, all kinds of birds, including the wild birds, are in these markets. In the sign, for those that can read Chinese, it says, be careful, the floors are very slippery. It's not just because they're wet either. The Hong Kong folks stamped that virus out by killing all of the poultry. And so nothing much more happened until 2004. But it came back from the ducks and the geese in southern China. Then in May 2000, it started spreading to chickens and people. And in 2005, in May, it got back into wild birds, and the wild birds carried it to Europe, Africa, India, and so on. Currently, these are the figures, and it is still a problem. It's noteworthy that the virus hasn't managed to get to the Americas. We'll come back to that later. So what's going on currently? It's gone off the pages of the newspapers. There's not much interest in it anymore. It can't be a problem. It's still a problem. In fact, it's an increasing problem. These are the countries affected. In 2010, the red is domestic poultry. The green is the wild birds. The three main problem areas, Southeast Asia, Vietnam, this whole block of countries, another group here in Indonesia, and in Egypt. So those are the three places that we have great problems at the moment. And every other day, a human dies. Direct contact with poultry, but it doesn't spread human to human. So these are the figures. The epidemic curve, if you like, was starting to come down in 2008. Then when the center in Egypt appeared, it's back up again, and it's not improving in 2010. Still not as many people dying, perhaps becoming milder, but we believe that in Egypt, children are diagnosed very, very early, and which doesn't occur in some of the other countries. So let's turn to Egypt. How did this develop in Egypt into a pandemic center? The virus came in March 2006. Really, a single virus came in. We call it Clade 2.1. And overall, there have been 106 infections, 32 fatalities, still going well in Egypt, and has not been stamped out. In July 2008, it was declared endemic in domestic poultry. So that it's just everywhere. And so with the advice of FAO, WHO, and OIE, they advised the Egyptians to use vaccine in the poultry to try to contain it. They bought up as much vaccine as they could obtain from Europe, China, elsewhere, tried the vaccines in Egypt, none of them worked. They tried as many as 3 doses in the same chicken 3 times. They failed, and that surprised me. So I had the same vaccine sent to us in the United States and tried them in United States chickens. They all worked. And so why did they fail in Egypt when the same breeds of chickens were being used? And after considerable amount of work, we discovered that the maternal antibody that is in the day-old chicken inhibits the generation of the immune response. We get suppression of the immune response in young chickens. And that mechanism we don't fully understand. And so we're currently working on different strategies to get around that problem. Moving to human vaccines. Human vaccines aren't very popular, but at my age, I take it every year, and they really do work. So what about human vaccines? They're mainly egg-grown. They're inactivated subunits, and some are non-adjuvated, which are universally available. Then the other universal vaccine is with alum adjuvant. And then there are other vaccines, the H1N1 pandemic vaccines with these commercial adjuvants, which are really shark oil, squalene-based shark oil adjuvants. Adjuvants potentiate the immune response. These are available in the European Union, but not USA. USA is extremely nervous about adjuvants. On the other hand, the live attenuated vaccines, which are really quite effective, are useful in the United States, and Europe is extremely nervous about them. So there's lots of nervousness about flu vaccines. What are the problems? Eggs are old kind of strategy, and they're not so wonderfully pure. The adjuvants, the Americans don't trust them, they're not sure about them, and it really takes too long. 6 months, the time the vaccines were made for the pandemic, the virus was all through the southern hemisphere, and it's simply 6 months is too long. And some of the things that happened, we just simply don't understand. We couldn't use our reverse genetic systems to make this virus. We don't know why. There are many improvements that should be made in preparing vaccines. Millions and millions of doses were prepared in the United States. There's hundreds of millions of doses sitting unused. Less in Britain and Europe. So why people don't they use these vaccines? Are they worried about safety? Autism is one of those bugaboos that scientifically we've shown that it doesn't apply. The public don't believe us fully. Thimerosal, the mercury preservative, is mainly removed from the vaccines. The old 1976 H1 vaccines was associated with ascending paralysis of Guillain-Barré. But the public, by and large, are still very nervous about flu vaccine. I think the real reason why there's so much unused vaccine was that the virus didn't kill enough people. If it killed more people, everyone would have been screaming for a vaccine. And so we have these vaccines on the shelf. The other strategy for control are the antivirals. The antiviral to the ion channel are the adamantane anti-influenza drugs. To the neuraminidase, the one that Graham Laver was involved with, blocks the active site of the neuraminidase. The first of these was zanamivir, the other is oseltamivir, Tamiflu. And so these two families, the Simitrel, the amantadine families that target the ion channel, is a very good drug, but we get very, very rapid resistance. The other oral, or Tamiflu is an orally administered drug. Relenza is inhaled. So the most often used is Tamiflu. What are the problems with the antivirals? Resistance. These, when we use single drugs, as we've learned from HIV, as we learned from cancer, you use a single drug, you will get resistance. These are RNA viruses, you are going to get resistance. The seasonal H1N1 viruses that have mainly disappeared became completely resistant. The pandemic virus is resistant to amantadine that was brought across from the pigs. So these are the troubles. We're mainly using one drug. This drug doesn't work well enough. But even these two drugs have not been combined commercially, which is a terrible mistake. The strategies that we have to work on is at least two drugs against different targets. Drug 1 affecting the neuraminidase, drug 2 working on the ion channel. This is the most simple-minded, should be done. We need new drugs. The other strategy is to have one drug against the virus, one drug treating the immune modulation of the human, mainly aimed at reducing the emergence of resistance. So returning to the pandemic of the past year, overall, we have been extremely fortunate. This has been mild. We started off thinking it was going to be like 1918, and we simply can't tell, based on the sequence of the virus, based on the biology, what we can do, we can't predict severity. And so we have to just work through it. And it's quite clear that overall this is, even if structurally very much the same as 1918, it's still rather mild. But still, we have to keep in mind, it's killed 18,000 people in the world and made people very sick. Affected the same age group, interestingly. 5 to 49 years were most affected. And it did cause problems in intensive cares throughout the world. A big problem in pregnant women. Interestingly, a big problem in obese people. Very noticeable in the United States. And my colleagues have shown that it's also, if we have fat mice, we can show that this virus kills them much more quickly. So that we can start to understand what's going on. The virus, the 2009 virus, is currently antigenically stable, which would suggest we're not going to have to make immediate changes. But don't trust it, because it's out there still reassorting. It's resistant to one of our antiviral drugs already, and we are finding oseltamivir resistance appearing now, hundreds of cases of resistance. But again, these are not transmitting yet. Give it time, it will. So what are the current issues with seasonal pandemic influenza? Will the pandemic H1N1 replace the last seasonal? It looks as though it's going to. It's already well advanced in replacing the older H1N1. Will resistance to the H1N1 be acquired from the oseltamivir resistant H1N1 that was circulating. If that doesn't occur, it'll occur by mutation. The one that worries me most, will that virus reassort with the H5N1 virus that is so pathogenic? So the gaps in our surveillance in 2010, we were not doing prospective surveillance in apparently healthy pigs. The surveillance is still rather weak. There's a resistance to surveillance in healthy pigs. My agricultural colleagues say, oh, but we do surveillance in sick pigs. That's true, we do, but we want to know what's going on in the healthy pigs out there in the world. And so maybe we need to put into place a global network similar to the human network. It's not a big deal to do that. The other is whether or not this H5N1 killer virus has got into the wild birds of the world. It does, it goes into the wild birds, but is it being perpetuated through the breeding sites into the next generation of wild birds? I hope not. I don't believe it has. Otherwise, it should have spread to the United States and the Americas. But there is concern with viruses that have spread recently into Bulgaria. There's a strain of H5N1 called clade 2.1.2 that seems to have got stuck in wild birds. So we're going to have to watch that. I want to finish up by spending a little time with my friends, the duck. The duck is, to me, and Ida Askunas, when I worked with her a number of years ago, she said to me, Rob, you've got to understand why the duck is more resistant than the chicken. And I've had postdocs trying to work with this. They start working with the ducks and they say, oh, there's no reagents. They go back to the mice. But recently, we've managed to get some that will work on the duck. And the duck, you know, you've all heard about the Trojan horse. You know, the Trojan horse in Troy with the soldiers inside. The duck, the wild duck, is the Trojan horse of flu, particularly this H5N1. Because it gets the virus, it poops it out, but it doesn't die. If it died, it wouldn't be a problem. So this is the problem of, it's good that the duck doesn't die, but that is a problem. So why is the duck resistant? My students from Canada, they've been working with me recently, published recently. They showed the duck possesses an influenza sensor, a sensor for influenza called rig-eye. Somewhere in the development of poultry, the chicken, including the wild chicken in Borneo, lost rig-eye. And so it's sensitive to the highly pathogenic viruses. And you put the same virus into a duck, it grins and kills the chicken. Rig-eye initiates production of interferon beta, which leads to activation of the innate immunity. And so that's what rig-eye does. My students keep talking about transferring the duck rig-eye. Well, they transfer the rig-eye of ducks to chicken cells, and then the chicken cells turn on the antiviral response. And now they want to move the rig-eye into domestic chickens to make them resistant. And there are pros and cons. Then you'll have Trojan chickens that don't get sick. So, you know, it's... but we are starting to, we are starting to understand something about the reason why ducks are resistant. And so this is my second last slide, and this is about where I come down with the present situation with flu. The riders of the apocalypse are saying, looking behind to the bird flu, that guy gives me the creeps. And so it does. This virus, as long as it's out there, we have to remember that the swine influenza virus from 1918 to 1998 didn't do very much, except it's got something in 98 that allowed it to reassort. This H5N1 is really only playing games with mammals since 1997. But we can't seem to get rid of it. One of the important things that FAO and OIE have got to do is to get this virus out of the chicken populations and duck populations of the world. Otherwise, we could get into real trouble. So what about the future? Better surveillance, combinations of drugs that we need, pre-pandemic vaccines. I'm not sure how well they're going to be accepted and whether we can get it right. What we can do is speed up vaccine production. We can sequence one of these viruses in a matter of hours. We can synthesize antigens. So why can't we start and think about a different strategy for making vaccines? And what we have to do is to improve vaccine distribution to developing countries of the world. Many countries still haven't received vaccine, and that's just not appropriate. And so I want to conclude by dedicating this lecture to Graham. He was a great scientist, but a rat bag. I had the honor, if you like, of writing his memoirs for the Royal Society. And I discovered that his forefathers came from London and they were shipped free out to Australia. And so maybe that's some of the reason why Graham was such a character. So but before concluding, I'd like to acknowledge my institution, St. Jude Children's Research Hospital, the National Institutes of Health, LSAC, that provides a lot of the infrastructure, the kinds of containment laboratories we need, and the many people, Elaine Tuaneman, whose department we work in and supports us, Richard Webby, Charlie Russell, Stacey Schultz-Cherry, Paul Thomas, Elena Gorbakova, Subrata Barman, Jerry Aldridge, my colleagues in Hong Kong, Iguan, Malik Pieris, Gavin Smith, Leo Poon, K.Y. Ewan, Honglin Chen, and Kathy Magor and Megan Barber, who did the RIG-I work, and the many WHO collaborating centers in Egypt, Vietnam, Indonesia, Thailand. And now, before I conclude, I also want to thank my daughter Sally at the age of 10, who went to the Barrier Reef also and caught those birds out of burrows in the sand, and my wife Marjorie for putting up with me running all over the world for most of my life. Thank you for your attention.
Excellent. You take questions?
Sure.
All right. Professor Webster will now take questions. We have two microphones, one on each side of the room. So if you'd like to ask a question, put your hand up, and then somebody else also keep their hand up so we can get one microphone to one and one to the other. So the first question here, and then the second one over there.
By what method do viral traits spread from one strain of the virus to the other strain of the virus? Because I thought that they worked like bacteriums. The offspring viruses have roughly the same genome as the as the virus particle that there was their parent, and they do not get traits from any other virus particles.
Can we get someone with a bigger voice? I'm afraid my hearing's down a little bit, and
I can't hear it either. Sorry.
just shorten
Tony,
it.
did you hear the question? Yell.
So how does a virus get a trait that was not present in its parent virus particle? How do two different strains of a virus combine their traits?
How does a virus get a new trait that it doesn't have? Is that the question? How does a virus get a new trait that it itself doesn't have? How does it learn to spread? How does it learn to replicate in the respiratory tract? It has two strategies. The influenza viruses are RNA viruses, and so they're constantly making mistakes as they replicate. And so the errors that they make are not corrected. And if those errors function in the virus, then it can give advantages. I compare it a little bit to the production line for a Chevrolet in the United States, where the workers have been out the night before drinking all night, and the next day they just throw the pieces in. 99% of those cars won't work. But then just one occasionally you'll be a winner. And this is how the virus obtains new information, mutations that give a selective advantage. And that's the other strategy it uses is to reassort with another virus, to mate with another virus, and bring in a gene that, when it's put with the other genes, will make it multiply more. It will replicate. Viruses like to replicate in the bird at high temperature. That lets the polymerases work faster. If you move those over into humans, it replicates faster, grows higher, and may be one of the traits that allows it to spread faster.
Okay, for now, one. Yes, thank you.
Hi. You mentioned in your lecture that there's difficulty in extrapolating from the whole genome sequence to how pathogenic the virus is, and that in the recreation of the 1918 pandemic flu, that they'd shown that that was related to very high cytokine levels. Has that been done for other flu viruses, or are there any plans to do that for the recent pandemic flu virus? And if we were to utilize that approach, would it be possible to predict how pathogenic future viruses would be? Did you hear that?
Yeah. Could you just summarize that and speak a bit more slowly? We're both having problems tonight for different reasons.
Okay, so my question was related to extrapolating between the gene sequence, whole genome sequence of the virus, and pathogenicity. You said that there was often a poor relation between the two. I was particularly interested by your comments about recreating the Spanish 1918 pandemic virus, and that had been shown to be related to very high cytokine levels in the animal model. Are there any plans to replicate those experiments for the recent pandemic flu? And would that provide a model to determine what future viruses we should be worried about in terms of pathogenicity?
Yes, I thank you for the question. The 1918 and the cytokine storm, as we talk about, and is it occurring with other viruses? Yes, indeed. The H5N1 viruses, they do the same kind of cytokine storm in mice, ferrets, and monkeys. And so one of the strategies that we're thinking about is to modulate the cytokine storm, because both the 1918 and the H5N1, it's the body that's killing itself by overproduction of cytokines, out of regulation cytokine responses. And so that's the other thing that we're doing at the moment, to try to damp down these responses. We can't shut them right off, otherwise we shut down the immune responses. So we're having to come up with drugs that will attenuate the cytokines but not shut them off.
Okay, thanks. One here, and then one in the back, but one row. And slowly and loudly, please.
Hello. Thank you very much for all your informative lecturing tonight. I used to live next to Jonas Salk, Dr. Jonas Salk, who didn't consider himself a world immunologist, but most people would have with all the work he did for polio. Not only was he brilliant in my view, but humorous. He used to call La Jolla BBLJ, beautiful, boring La Jolla. And he thought that helped our own immunology, which gets to my question. Jonas Salk took handfuls of vitamins. He drank wheatgrass every day. If you were lucky enough that he ever cooked for you, you would see that he believed in all that. Vegetarianism, major vitamins. He didn't like cats ever, and he wasn't much better around dogs. And very likable guy, really. He considered himself a hermit because it took many years in the lab to get that polio vaccine. But in your opinion, and you're very brilliant yourself in my view, how much can we up our immune system by those things that Salk was trying to do? The rest, the vitamins, the food. And the last question I have is, he prevented a lot of trips because he wouldn't take, he didn't like the malaria vaccine, so he wouldn't go to India. He wanted to, but he wouldn't go because he couldn't decide whether he feared the disease more or the vaccination. So how do you feel in general today about the various vaccines? But first, how do we up our immune system? Thank you.
How do we up our immune system? Well, besides using vaccines, the way to up the immune system is high-quality, non-reactive, safe vaccines. That's the ultimate way. If you want to...
Not the wheatgrass then, huh?
But on the other hand, you've got to be healthy, you've got to have enough vitamin C, and otherwise the immune system can't function. But, you know, when we go back to 1918, and in the strategy for treating influenza then was zinc sulfate. You sprayed the people with zinc sulfate. We still sell zinc tablets for treating flu, and it's supposed to upgrade the immune system and make us strong. Well, if you believe that, then it will perhaps, but it really doesn't.
The other part, how do you feel about vaccines?
Sorry?
How do you feel about vaccines?
How do I feel about vaccines?
I think you answered that one for us.
I take vaccine every year. I think that we can improve these vaccines. That's my take on vaccines, is we can produce better vaccines with better adjuvants that are perfectly safe, faster.
Cats and dogs? Any fear of cats and dogs like Jonas Salk had?
Are you afraid of cats and dogs? Do you avoid cats and dogs?
No, I have cats.
Right. 2 rows from the back. Thank you for being patient.
To persuade our political masters to fund better surveillance in pigs and in the avian population?
What would be our strategy for persuading our political masters for better surveillance of animal populations?
We keep working and trying to persuade the folks to do better surveillance in the animal population. I think there is good surveillance in the avians of the world, the wild birds of the world. There's enormous surveillance going on now. Domestic surveillance is modulated by trade. If you have these highly pathogenic viruses and are exporting chicken, sometimes some countries really don't want you to know. And so it's kept a little bit under the counter. But by and large, avian surveillance is good. Pig surveillance of healthy pigs is less than optimal. That's where we as scientists have to convince the pork boards of the world that they won't lose their livelihood if we find viruses in them. And we will. So we have to do some work in public relations to get these things on board. Can be done.
Thank you. And another one at the back there, yes. And then one this side, next.
At the beginning of the lecture, you gave a disappointing score of 4-0 to the virus. I'm wondering if perhaps the scientists could be awarded 1 goal for Tamiflu and Relenza.
I would agree with that entirely. I think that the structural biologists, the work that John Scale has done, that Labour did, and the work that's going on in the polymerases, we just need more of it. But on the other hand, this virus is so smart. Its strategy, we all planned for the next pandemic, and none of us, not the very best people in the world, dreamed that H1N1 would reappear. And so we just, but I'd give them 1.
And yes, halfway down here, hand up, white top. Thanks.
I'm a medical student, and I've spent some time with some community nurses who were quite anti the swine flu vaccine. They thought it had been produced too quickly and were in fact telling patients that they wouldn't have it. So I have 2 questions. First of all, was it produced too quickly? And secondly, what can we do to convince healthcare professionals that the vaccines are safe and to not convince patients not to have them?
I think you're asking exactly the same question as me. Why do people distrust vaccines? But because we haven't got it completely right yet. We keep getting some incidents of reactivity with vaccines. With the swine flu vaccine, there has been some cases of reactivity. And we have to live with the possibility that there are some people who are going to react. But the risk-benefit ratio is so much in the favor of the use of vaccines that we should use them. We have the same problem at St. Jude. There's cancer hospital, and we have to persuade the staff quite strongly that they've all got to be vaccinated because the cancer kids at St. Jude will die if they get flu and mess up their transplants. So this is a problem that none of us have solved. And some of the worst people at taking vaccines are the young physicians. Sorry about that.
Okay, this may be, 2 questions then, and then my time is up that I'm allowed to spend on questions. So 1 here, and, maybe 3 then. 1 here, 1 there, and then 1 quickly at the end. Okay, so 3 more questions.
Thank you for a fascinating lecture. Can I ask you to talk a little bit more about the periodicity of influenza pandemics? We used to be told that we could, We used to be told that you could expect influenza pandemics about once every 10 years, but the interval between the last pandemic and this one has been something like 41 years, which I believe is the longest since 1849 to 1889. Is that a sign that we're doing something well, or is this, in your view, pure happenstance?
In my view, it's pure happenstance. We based the idea of 10 years on the Asian and the Hong Kong, and there were too few. A sample of 2 is not big enough. I think that's the answer.
Okay.
Do you think that in the future, like nanotechnologies will have a role to play in fighting viruses. Possibly in like the production of vaccines, but maybe in like directly preventing viruses sort of infecting people. I don't have any experience particularly of it, so it's quite an open question.
My knowledge of nanotechnology is not as much as I'd like to have, but instinctively, I would say yes, it will have an impact, but don't ask me where.
Then I think the last one here.
I've made the observation that in the U.S., pork has to be raised on concrete, and the reason for that is prevention of parasite vectoring. But I wondered if anybody, usually those facilities are indoors, so the hogs are raised indoors. And I wonder if anybody has looked at breaking the wild bird swine people host vector cycle through isolation. Isolating the hogs from the wild birds.
Yeah. Could you break the cycle by isolating the sick birds from the well birds?
Well,
the birds, the wild birds
isolating
from
the pigs and the birds and
sick animals from the wild.
Yes. There is no doubt that this is going on in the raising of poultry in closed sheds. But there is a feeling that we would like to see chickens raised naturally. In fact, one of my pet peeves going on in the United States at the moment is that backyard chicken raising. I went to fill my car with gasoline 2 days ago, or before I came early in the morning, and there was a rooster downtown sitting on the fence crowing. And so there's a big movement of people bringing chickens back into their backyards. This was the problem in Egypt. And so you're right, we have to separate these species, and whether it's a good idea to introduce additional pigs or chickens in close contact with humans is the problem. The backyard poultry are the big problem in Thailand at the moment. And so you point to what we should do. It's not easy to achieve because the many poor people in the world depend on those backyard poultry.
Right, thank you. I think we should call it a day there. I want to thank Professor Webster on your behalf for a very interesting lecture. I don't know, I learned a lot. I don't know whether I am now encouraged or terrified after listening to what he had to say. I was sort of rather complacent, I suppose, but we ought to be cheered, but at least somebody has got a grip of what's going on, the statistics and what might be happening. But I'm not thinking of raising chickens in the near future in my backyard. So we come to the end of the lecture. If I'm right, there are some things here which I have to present Professor Webster with. First of all, a certificate, another one to match the one you had in 1989, a box to match that, and a medal, the Leeuwenhoek medal, rather fine. Okay. And a check, and I have no idea how much for, but a check's always good news. Okay, thanks very much.
Thank you.
Thanks very much indeed. Terrific lecture.
Thank you.
Now you can have